Hi, hello, namaste. Welcome to Shaf's Biotech for you. We would now focus on tools of biotechnology. Today we'll discuss about restriction enzymes or restriction endonucleases. Restriction enzymes are the enzymes that recognize a specific base sequence in DNA and make a cut on both the strands. These are also called as molecular scissors. The first restriction enzyme to be isolated was HIND2. This was isolated from Haemophilus influenza by Werner Arber, Hamilton O. Smith, and Daniel Nathans in late 1960s and early 1970s. The specific nucleotide sequence or the short nucleotide sequence specific for each restriction enzyme is called as recognition site, and the process by which a restriction enzyme cuts the DNA is called as digestion. When a restriction enzyme recognizes a sequence, it snips through the DNA molecule by catalyzing the hydrolysis of the bond between the two adjacent nucleotides. Hydrolysis is nothing but splitting of the chemical bond by the addition of water molecule. The digestion process is of two types. One, blunt end digestion. The example of the restriction enzyme that carries out this blunt end digestion is HIND2. As we can see, here is a DNA molecule with the ATGC sequence. HIND2 recognizes a sequence between GC. This is a particular recognition site for HIND2. Within the recognition site, it makes an equal cut, or it makes a cut at the same position on the two strands, resulting in blunt ends. Coming to the second type of digestion, that is staggered, sticky end, or cohesive end digestion. The example of the restriction enzyme that carries out this kind of digestion is EcoR1. EcoR1 is the restriction enzyme isolated from E. coli strain RY13. Here, as we can see, this is the recognition sequence for EcoR1 or the recognition site for EcoR1 restriction enzyme. EcoR1 cuts the DNA after guanine on both the strands which are not at the same position, resulting in the staggered or the sticky ends. Coming to the types of restriction enzymes. There are four types of restriction enzymes. The first one, or the type 1 restriction, they cut far from the recognition site. They require both ATP and S-adenosyl methionine, and they have both restriction and methylase activities. The second type, or the type 2, they cut within or at the recognition site. They require magnesium for their function, and they have only single function which is independent of methylase. Type 3, they cut at short distance from the recognition site, and they require ATP, but they do not hydrolyze this ATP. The presence of S-adenosyl methionine stimulates the function, but it is not a compulsory component. They are part of a complex with a modification methylase. Type 4, they target modified DNA. Example of modified DNA is hydroxymethylated DNA. With this, I wrap up today's session regarding the restriction enzymes or endonucleases. Please like, share and subscribe to my channel.